you. All right, so my name is Shalin Naik. I'm group leader at the Walter and Eliza Hall Institute, now known as WEHI. And I'm pleased to talk to you about a story that just came out in Nature Immunology a couple of days ago. So it's quite fresh, which is good. And here's the article. It's identification of a new lymphoid-primed progenitor marked by Dach1 downregulation. So in classical models of haematopoiesis, we have situation where we've got long-term stem cells, short-term stem cells, this multipotent progenitor called the LMPP, and then we have these series of ball and stick type diagrams which describe how a stem cell gives rise to the diversity of immune cell types. And this is a figure from a review I wrote. It's very dendritic cell centric because that's a major interest of our lab, but the principles are very similar. An LMPP is a progenitor that we were quite interested in studying back during my postdoc, and that was one identified by Sten Erik Jacobson's group in 2005, which they proposed was a common B and myeloid progenitor, but not megakaryocyte progenitor. And taking this population and using a cellular barcoding technology, when I was in Amsterdam, we labeled individual cells with DNA barcodes. We put them into a mouse and then let those progenitors divide and develop as they otherwise would into the different lineages. And in that way, we found that multipotent progenitors were actually not very multipotent at all. And as you can see here, the proportion of LMPPs that generated all lineages was actually very small, and they were otherwise biased towards B cell development, dendritic cell development, myeloid cell development, or combinations thereof. So we were interested in, well, how does one prospectively isolate progenitors that are primed for lymphoid, dendritic cell, and myeloid? To frame it a different way, can we put these in different buckets? Now, the answer for dendritic cells actually came in 2019 from Tamura's lab in Japan, where he found that an IRF8 GFP positive subfraction of LMPPs was highly biased to CDC production. But there was a question mark around whether there were, in fact, lymphoid-primed, lymphoid multipotent progenitors and myeloid-primed progenitors. And so we answered this question with single-cell RNA-seq. So in 2013, we Googled single-cell RNA-seq. CellSeq was the top hit. So we bought all the reagents. We did our first experiment and we adapted it to a version that's very similar to CellSeq 2. And we got our first data in 2014. And that's actually figure 1. It's only 192 cells, which is laughable by today's standards. But actually, that was perfectly fine for us to see clusters of heterogeneity that when we looked a little bit deeply, we found there was this lymphoid-like cluster and this myeloid-like cluster. And interestingly, both of those clusters of cells fit into what was previously described as this LMPP, or otherwise known as MPP4 gate. Okay, so these LMPPs or MPP4s seem to be heterogeneous, at least by single-cell RNA-seq. And the great thing about CellSeq and plate-based methods is you have the FACS markers at the time of sorting, so you can do this sort of backgating analysis onto flow cytometry. Okay, and we searched for a bunch of surface markers, we tried a few, didn't really find a way to separate them, and then turned towards more intracellular markers or genes, in this case, Dach1, which was an understudied transcription factor in haematopoiesis. I'll tell you straight up, unfortunately, when we knocked out Dach1 using CRISPR and in transplantation assays, we didn't see any difference in the development of myeloid cell types, lymphoid cell types at multiple time points all the way out till day 91. So unfortunately, in this system, Dach1 knockout alone does not have an effect on any of the lineages that we tested. Okay, but does Dach1 maybe demarcate this heterogeneity? So by complete coincidence, there was a Dach1 GFP reporter mouse that had been made by the Hilton and Alexander groups in the lab next door to us. So we took this mouse and we fractionated the population. We looked at the LSK fractions that were, then we split the IL-7 receptor positive into CLP, common lymphoid progenitors. The IL-7 receptor negative, we split into long-term HCC, short-term HCC, MPP 3, 4, and 3 and 4, otherwise known as LMPP. And when we looked at the expression of GFP on this mouse, sorry for the low resolution here, but we found that long-term, short-term stem cells were very high for Dach1 expression. Dach1 GFP high. CLPs, on the other hand, were Dach1 GFP negative. And what we found was that LMPPs were heterogeneous for Dach1 expression. So then we did the reverse experiments. Like, now that we've discovered Dach1 might separate progenitors, we then had to ask, are actually Dach1 low MPP4s actually transcriptionally homogeneous and potentially lymphoid primed? So we took, now instead of black 6 mice, we took the Dach1 GFP mouse, we performed index sorting of our stem and progenitor cell fraction with various surface markers at the time of FACS sorting so that we could pull those populations back out later. And we found, long story short, that indeed we could find a putative, what we called lymphoid primed progenitor, because it was IL-7 receptor negative, so it's not a CLP. It was FLT3 positive, as we know MPP4s are, but it wasn't the only population that was FLT3 positive. So as you can see here, there's this big cluster of FLT3 positive cells, some of which are high, that are different from this population. But importantly, this fraction was Dach1 GFP low, and it was transcriptionally homogeneous, and it contained genes that were sort of indicative of a lymphoid kind of origin. Now, we aren't the first to have found this transcriptional cluster, and almost every single cell RNA-seq data set you see that has been published in the last few years kind of sees this branch of lymphoid development. But no one had really gone and validated whether that actually is lymphoid primed or not. So just to kind of complete this section, now looking at a Tisney plot of this HSPC fraction, this red cluster is what we call putative lymphoid primed progenitors. And they were very distinct transcriptionally to genuine common lymphoid progenitors, as you can see here in this brown cluster. Common lymphoid progenitors are FLT3 positive, c-KIT a bit lower, IL-7 receptor positive, but they're quite transcriptionally distinct here. And here and now, of this putative LPP, we can see which genes are expressed in that cluster. So DNTT, which is otherwise known as TDT, which is the machinery for T-cell receptor diversity. SATB1 was quite highly expressed, Notch1, but not, for example, V pre-beta 1, which is this silent transcript that's present in common lymphoid progenitors. And now that's based on gene expression, but of course we also have the surface markers. So indeed, this population, CD150 negative, CD135 positive, Dach1 GFP negative, but largely IL-7 receptor negative here. Okay, so this looks like something different. What can we do about it? Well, we can, take the old school LMPP gate, which would be the top 25% of FLT3 expressing cells. So in this case, it's not the top 25%, it's the top 40%. But we took a slightly liberal gate here just to demonstrate that if we take these Dach1 positive LMPPs, we find that actually they don't contain many cells at all in this putative lymphoid prime progenitor transcriptional cluster. So these cells by FACS do not fall in the transcriptional cluster of these LPPs. By contrast, the LPPs that we're calling them, these red cells here, did largely enrich in this lymphoid prime cluster, but there were some cells that fell outside of this as well. And this is in contrast to gated IL-7 receptor positive, FLT3 positive common lymphoid progenitors. They tended to be really at this apex, although there were a few that sat in the top of this apex here. Okay, so from this, we proposed hypothesis 1 that putative lymphoid prime progenitors are a subset of LMPPs, will be lymphoid primed, more so than lymphoid primed multipotent progenitors. So how do we test this? Oh yeah, and sorry, the second hypothesis when we did trajectory analysis was that These LPPs, which are here in this red fraction here, precede the brown cells here, which are common lymphoid progenitors. So our hypothesis was, one, that the LPPs are lymphoid prime, but two, that they give rise to lymphoid cells via a CLP intermediate. Okay, yeah, so that's written here. Hypothesis 2, LPPs give rise to CLPs en route to lymphoid generation. So let's first validate hypothesis 2. Do LPPs generate CLPs? So we transplanted either LPPs or Dach1 positive LMPPs into partially irradiated mice, and we analyzed their lineage fate at multiple time points across time. And here's day 7 in the bone marrow of these mice. And you can see that while Dach1 positive LMPPs are largely myeloid, that is CD1632 progenitor generating, the lymphoid prime progenitors, LPPs, don't generate many IL-7 receptor positive cells, which are indicative of CLP generation. By contrast, it's the LPPs gave rise to a greater proportion of IL-7 receptor positives compared to CD1632 positives. And if we measure that output, multiple mice, multiple experiments, you do see that the LPPs are generally greater bias towards LPP generation rather than myeloid progenitor generation, and vice versa for the Dach1 positive LMPPs. So LPPs give rise to CLPs, more so than Dach1 positive. All right, validation of hypothesis 1. Do LPPs lack myeloid potential in vivo? So we took this, we took a broad gate of CD135, also known as FLT3, and then in this fraction, we split our cells into Dach1 positive LMPPs and the Dach1 negative LPPs. I won't show you this data today, but it's in the paper. We also split this same fraction by FLT3 very high and FLT3 high. So FLT3 high versus very high, and transferred those as well. And when we transferred the FLT3 fractions, we saw myeloid generation from both of these. There was a slight enrichment for lymphoid generation, but not statistically significant. So FLT3 is not sufficient to separate lymphoid generation from myeloid generation, even when you take the very high fraction of that. By contrast, when we take a Dach1 based separation of LMPPs, now this is the top 25% here, we find that the lymphoid prime progenitors generate T cells quite early after transfer. So already at day 28, we're getting CD4 and CD8 T cells, and we're already getting B cells at 2 weeks post-transplantation. This is in contrast to Dach1 positive LMPPs, which can generate T cells and B cells, but really not until day 52 post-transplantation do you get lymphoid cell generation. So we think that the Dach1 positives can give rise to LPPs and then give rise to CLPs and lymphoid cells, but they're a step behind. And I'll show you the model at the end that might answer that. By contrast, LPPs could not give rise to eosinophils, monocytes, or neutrophils at any time point tested. And we did this both in vivo and in colony assays in vitro. And really, they are lymphoid restricted, just no monocytes, no neutrophils, no eosinophils. So now I think we can definitively call them LPPs because we functionally tested them. All right, so we have now, with LMPP heterogeneity, we've now added, well, I'm going to call this a DPP, a dendritic cell prime progenitor, because I can. And I'm going to call these now what we've discovered today, lymphoid prime progenitors. And so the question is, does a myeloid prime progenitor exist in this fraction or not? Or are the Dach1 positive LMPPs still heterogeneous in their fate? We've got barcoding experiments ongoing to try and answer some of these questions. But for now, I will now, for the last minute, talk to you about how this fits in the models of hematopoiesis. So many of you will be aware that these classic models of hematopoiesis, the one I started with, are now kind of being rewritten in light of the new single cell RNA-seq data and the lineage tracing data that is emerging. And so this is probably a more refined way of answering this, of modeling what hematopoiesis looks like. And this is my version, which you can find in this review I wrote this year, where there's actually streams or trajectories of development where cells already in the long-term stem cell fraction, if they're on this side, they can already be myeloid primed, or if they're on this side, they can already be lymphoid primed. And as you move down the hierarchy, these classic boxes that we draw as gates by FACS are actually encompassing quite some lineage priming heterogeneity. So as you see here, this box specifies the LMPP, but actually within the LMPP, you have, as we've shown with barcoding and now with discrete markers, different fractions of commitment. And so here you've got the DC prime progenitors, but now we can add lymphoid prime progenitors to this tree here. So we're fractionating the LMPPs further. Oh yeah, this is an old slide. It's no longer in press. And this is actually another one of my favorite representations from my friend Martin Guillaume and his co-authors on this manuscript. I think it nicely describes how commitment can be already embedded a little bit, and what you see is an emergence of that phenotype as you move down that hierarchy. All right, with that, I would like to thank Daniela, who really powered the single-cell RNA-seq development and did the first experiments of those 192 cells all those years ago. Lu-Yi Tan, who developed all the bioinformatics and analyzed that data. Yaring, who did the very first biological validation experiments, and Sarah, who really finished them off. And plus all my collaborators in this paper, and NHMRC for funding the work. So with that, I will take questions and see how Professor Tao is, whether he's available to take these questions. If not, I'll take those questions. Hi, Professor Tao.
Yeah, I'm back.
Hey, you're back. Welcome.
Yeah, I'm a little confused. Who is going to ask your question? It's just between you and me?
Yes, just you and me. We're chairing the session. So I can just read my own questions and answer them, or you can ask them for me.
I'm a little bit confused. I thought the audience, from other places also can ask questions.
Oh, yes, they can. They can,
yeah,
and they have. There is a Q&A panel here, so yeah.
Yeah, maybe I go ahead to ask you a couple questions.
Yes.
Okay. First of all, the LLP, when you do the limiting dilution, did you see what kind of efficiency you will see the lymphoid specific, potential in your hand? Yeah. Second question is, of course, always everybody would wonder, is there any human mimic and based on mouse study?
Yeah. So to the first question, we did do limiting dilution assays. We saw a slight enrichment for lymphoid activity in the LPPs. I think it was for, it was one of the lineages, B or T cells. We did both DL1s and OP9s. I can't remember which one. It wasn't a huge difference in precursor frequency, I have to say. But what we did do then was actual single cell culture assays. And what we found that the clonal burst, the clonal output of single LPPs was much higher than the Dach1 positive. So if you score wells just based on plus minus, the efficiency is not different. But if you score them based on clonal output, we saw a big difference. And the LPPs generated much larger clones of T cells in DL1 cultures and B cells in OP9 cultures.
I see.
Yeah. So I'm not sure if you can see this, Professor Tao, but there's also a Q&A panel where people ask questions. Yeah. So Ankur, thanks for your question. Does Dach1 bind to the promoter's genes responsible for lineage commitment or leads to chromatin remodeling? Interesting question. We haven't tested this directly, but there was a study, I'm forgetting the last author now, but they looked at the methylation map of progenitors, and Dach1 was associated with myeloid and stem genes rather than lymphoid genes. So we definitely think there is a role there, but we need to tease it apart. And if the knockout is not giving us a immediate phenotype, the question is it redundant? was our CRISPR transplantation assay the wrong assay? We don't know, but good question.